Hello students, I am VLNS lecturer in Biochemistry from Sri Venkateswara College of Arts and Sciences, which is affiliated to Adikavinana University, Rajahmundry. Now, I would like to discuss about second B.Sc fourth semester Biochemistry paper, that is intermediary metabolism. So, regarding this paper, we have seen first and second units, and we are seeing topics from the Unit-3. Let's continue with the topic urea cycle. Okay, so in the previous class we have seen the reactions of amino acids. So now we will see about the urea cycle. So let's see about this. As we discussed earlier, urea is an important end product in the amino acid or the protein metabolism. So here amino acid metabolism is nothing but it is also a protein metabolism. Why? Because proteins are nothing but the building blocks of amino acids. Okay, so this amino acid metabolism is not just an amino acid metabolism, it is also a part of protein metabolism. Okay, so here commonly urea is the end product to all this protein and amino acid metabolism. So here urea is the end product of the protein metabolism, and here urea accounts for 80 to 90% of the nitrogen containing substances excreted in urine. So whatever the nitrogen containing substances are present in our body, all those nitrogen containing substances, from those nitrogen containing substances 80 to 90% they'll be converted into urea. That means their 80% to 90% of the nitrogen containing substances end product is urea. Okay, and urea is synthesized in liver, and it is from liver it is transported to kidneys, and there with the help of nephrons it will be excreted. So here production of or the location for the urea synthesis is liver. Later this urea is transported to kidneys, and from there it is excreted through urine. Okay, here urea cycle is the first metabolic cycle that was elucidated by Hans Krebs and Kurt Hansliet. Okay, so here these are the two scientists, Hans Krebs and Kurt Hansliet. These are the two scientists who identified this urea cycle. That means they elucidated each and every step that is present in the urea metabolism, that is urea cycle. So each and every step was explained in detail by these two scientists, that is Hans Krebs and Kurt Hansliet, and that's why we can also call this urea cycle as Krebs Hansliet cycle. So here urea cycle or Hans Krebs Hansliet cycle is the same. And here urea has two amino groups, and from these two amino groups one is derived from NH3 and another group is derived from aspartate. So this NH2 C double bond over NH2 is the urea structure, and here the two NH2 groups, one is derived from the NH3 and another from the aspartate. And here carbon atom is supplied by CO2. So whatever the carbon C present in the urea is donated from the carbon dioxide. And if you see this urea cycle, majorly it will contain 5 steps. Okay, and this is a cyclic metabolic pathway. So if you see glycolysis, it is a linear metabolic pathway, whereas Krebs cycle is a cyclic process. So in the same manner, this urea is also a cyclic process. And as it contains 5 metabolic reactions which will be carried by 5 enzymes, and here the first 2 enzymes will be present in mitochondria and the remaining 3 enzymes will be present in cytosol. Okay, so the first step is synthesis of carbonyl phosphate. So the first step is synthesis of carbonyl phosphate. Here the enzyme which is responsible for the first reaction or the first intermediate formation in urea cycle is carbonyl phosphate synthase 1. Carbonyl phosphate synthase 1, and this is present in mitochondria. As said earlier, the first 2 enzymes which will be present in mitochondria and remaining 3 will be present in cytosol. So the first enzyme which is responsible for urea cycle is carbonyl phosphate synthase 1, which is present in mitochondria, and this enzyme carbonyl phosphate synthase 1, and this will catalyze the condensation of NH4 plus ion with CO2. So here NH4 plus will combine with CO2 and finally a carbonyl phosphate will be produced. So what happened here? NH4 ion and carbon dioxide, they both combine to form a compound called carbonyl phosphate, and the enzyme catalyzing this reaction is carbonyl phosphate synthase 1. Okay, and this step consumes 2 ATP molecules and irreversible, it is not reversible. And rate limiting, here rate limiting means it is also helpful in the regulation of this urea cycle. Okay, so the first step, synthesis of carbonyl phosphate from NH4 plus ion and CO2 by the action of the enzyme carbonyl phosphate synthase 1. So let's see the reaction. So this is the first step. So here carbon dioxide and NH4 plus ion, they are combining to form this carbonyl phosphate. And the enzyme responsible here is carbonyl phosphate synthase. And during the reaction, 2 ATP molecules were utilized. Why? Because phosphate will be donated here. So this carbonyl phosphate, no, so inorganic phosphate will be donated from the ATP. Okay, so this is the first step. And the second step, formation of citrulline. Formation of citrulline. Here citrulline is the next intermediate product. The citrulline is synthesized from carbonyl phosphate, and here the enzyme is ornithine transcarbamylase. So here along with this carbonyl phosphate, so for every reaction one substrate and enzyme are required. So here the carbonyl phosphate and enzyme ornithine carbamylase doesn't just enough for, these are not just enough for the synthesis of citrulline, and they require this ornithine. So carbonyl phosphate and ornithine will react to form the citrulline with the help of the enzyme ornithine transcarbamylase. Okay, and citrullin proceeds in this reaction is produced in this reaction is transported to cytosol by a transporter system. So as the first two enzymes responsible for this urea cycle are not present in the cytosol, these two reactions will be carried in the mitochondria. So here citrullin, which is produced in the mitochondria, it will move towards the cytosol for further reaction. So citrullin produced in this reaction, that is in the second reaction, is transported to cytosol by a transporter system. So far what kind of transport systems do we have seen? If you remember, carnitine transport system we have seen. Tell me in which cycle we have seen this carnitine transporter system? So here in the same manner another transport system is helpful in this citrullin formation. So this transport system is responsible for the transportation of the citrullin to the cytosol. Okay, so let's see the reaction. So if you see the reaction here, carbamyl phosphate and ornithine, they are combined to form this citrullin. This is the citrullin. NH2C double bond O, NH, CH2, 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 CH and H3 plus COO minus, which we can call it as citrullin. And if you see here carbamyl phosphate, what's its structure? NH2C double bond O, O, P double bond O, O minus, O minus. That means here NH2C double bond O has an addition of inorganic phosphate. Okay. So this is how citrullin is synthesized. And what is the enzyme here? Ornithine transcarbamyl is the enzyme which is converting this carbamyl phosphate and ornithine into citrullin. So let's see the third step. So obviously this third step will be occurring in the cytosol. Why? Because citrullin now moved to the cytosol from mitochondria. Okay, so let's see what is going to happen during this third step. And the third step is nothing but the synthesis of arginosuccinate. Arginosuccinate is produced during the third step of the this reaction urea cycle. Okay, now let's see. So here citrullin will condense with aspartate to produce this arginosuccinate with the help of the enzyme arginosuccinate synthase. So citrullin and aspartate will combine to form this arginosuccinate, and the enzyme responsible for this reaction is arginosuccinate synthase. And this reaction is a ATP dependent reaction. That means the enzyme arginosuccinate synthase, which is responsible for the production of arginosuccinate from the citrullin and aspartate, is a ATP dependent enzyme. So this step requires ATP, which is cleaved to AMP and inorganic phosphate. Later it is immediately broken down to inorganic phosphate. Okay. So here what happened in the third step? Citrullin is combined to aspartate to form arginosuccinate by the enzyme arginosuccinate synthase. So let's see the reaction. So if you see here, succinate by condensation of this aspartate, that is COO minus CH2 CHNH3 plus COO minus. This is the formula of aspartate, and this will combine with citrulline to form this arginosuccinate NH2 plus CHCOO minus CH2COO minus NHCH2CH2CH2 CHNH3 plus COO minus. So this is the arginosuccinate in the arginosuccinate structure, and here it is produced from the citrulline by the condensation of aspartate, and here the enzyme is arginosuccinase. Okay, succinase is the enzyme. So here this reaction occurred in cytosol, whereas the previous two reactions occurred in mitochondria. So now the second step in the cytosol and the okay, so here this is the first step in the mitochondria and the third step in the urea cycle. Now let's see the fourth step. What happened during the fourth step? So the fourth step is cleavage of arginosuccinate. So here citrulline and aspartate come in to form this arginosuccinate, and now again this arginosuccinate is cleaved. So arginosuccinase is the enzyme which will cleave arginosuccinate to give arginine and fumarate. So here the products arginine and fumarate will be produced, and this arginine is the immediate precursor for urea, and fumarate liberated here provides a connecting link with TCA cycle, gluconeogenesis, etc. So here whatever the this arginine, which is an immediate precursor for urea, so here what happens is, by reaction of this arginosuccinate into arginine and fumarate, arginine will be directly converted into urea, and fumarate will be liberated here by the involvement of this fumarate in the TCA cycle and gluconeogenesis. So let's see the reaction. So arginate is split into arginine and fumarate. So again by the enzyme arginosuccinase. Okay. So the fourth step is formation of urea. As in the discussed in the fourth step, this arginine which is produced from this arginosuccinate, this is the immediate precursor for urea. That means from this arginine only urea will be synthesized. So the fifth step is formation of urea. formation of urea. So here arginose is the fifth and final enzyme that cleaves arginine to yield urea and ornithine. Okay, so from this arginine, by the action of arginase, so arginine will be converted into urea and ornithine. So ornithine so regarded enters mitochondria for its reuse in the urea cycle, and arginose is mostly found in the liver, while the rest of the enzymes of urea cycle are also present in other tissues. Okay. So here now let's see about the regulation of urea cycle. So how we can regulate this urea cycle in case of higher amounts of production of this urea cycle may lead to some disadvantages, and for that reason there should be a regulating mechanism in urea cycle. So let's see about this regulation of urea cycle. So here the first reaction, as I said earlier, it is a rate limiting reaction, so which will help in the regulation of urea synthesis. So this is the first reaction, catalyzed by carbamyl phosphate synthase 1 is a rate limiting reaction and our committed step in urea synthesis. Okay, so first enzyme, that is carbamyl phosphate synthase 1, is a very important enzyme to convert this particular So here what happens, first enzyme, that is carbamyl phosphate synthase 1, will act as rate limiting reaction or committed step in urea cycle. So this enzyme carbamyl phosphate 1 phosphate synthase 1 is allosterically active by N-acyl glutamate. So by presence of this NAG, that is N-acetyl glutamate, this CSP1 is allosterically activated, and rate of urea synthesis in liver is correlated with the concentration of N-acetyl glutamate. So this N-acetyl glutamate is correlated with the rate of urea synthesis in liver. Okay, the high concentration of arginine increases NAG. NAG means this N-acetyl glutamate, and the consumption of protein-rich meal increases the level of energy in liver, leading to changed urea synthesis. So this is how urea cycle is inhibited. So we have got arginine and fumarate by the enzyme arginosuccinase, and now this arginine is converted into ornithine with the help of the enzyme arginase, and ornithine is converted into again this, it will move to the mitochondria where it will react with other compounds. That means here ornithine will be converted into citrullin by the addition of this carbamyl phosphate and ornithine with the enzyme ornithine transcarbamylase. Okay, so this is about urea cycle. Okay, so hope you understood well about this urea cycle, and now small let's see a small topic related to this amino acid metabolism. Okay, so based on the catabolism of the carbon skeleton of amino acids, we can distinguish these amino acids into glycogenic, and also the second one is ketogenic. So here let's separate these amino acids into glycogenic and ketogenic. So for that, let's see what are these glycogenic amino acids and ketogenic amino acids. So here The amino acids are classified into two groups based on nature of the metabolic end products of the carbon skeleton. So, if a amino acid undergone a reaction and it produced a carbohydrate product, it will produce a end product. Based on the type of end product, will distinguish this amino acid into a glycogenic or a ketogenic. Okay. So, let's see what are glycogenic amino acids first. These are the amino acids whose carbon skeleton is finally degraded to pyruvate or one of the intermediates of TCA cycle. So, the end product should be a it will be degraded from the pyruvate or any one of the intermediate of TCA cycle, that is Krebs cycle. And these intermediates serve as good substrates for gluconeogenesis, leading to the formation of the glucose or glycogen. So here these are the glycogenic amino acids whose carbon skeleton is finally degraded to pyruvate or to any intermediate of the TCA cycle. These will be converted into pyruvate or intermediate of a TCA cycle. It might be any intermediate. Okay. And the next one is ketogenic amino acid. Ketogenic amino acid. So, ketogenic amino acids are the amino acids whose carbon skeleton is metabolized to acetyl-CoA or acetoacetate, which can be converted into fat. So, here this acetyl-CoA or acetoacetate, particularly this acetyl-CoA is a TCA cycle product. So, as I said earlier, here this end product of ketogenic amino acids acetyl-CoA is one of the important intermediate in the Krebs cycle. Okay. So, some of the amino acids are both ketogenic and glycogenic. That means few amino acids show both ketogenic and glycogenic in nature. So, see here: alanine, cysteine, glycine, hydroxyproline, serine, aspargine, threonine, aspartate, phenylalanine, tyrosine, isoleucine, methionine, threonine, and valine. So, these are all few amino acids. So, here the amino acids leucine, lysine, phenylalanine, tyrosine and tryptophan, these are being converted into acetoacetate. So, here what we have seen, conversion of amino acid into acetyl-CoA or acetoacetate make it a ketogenic amino acid. So, here amino acids leucine, lysine, phenylalanine, tyrosine and tryptophan are ketogenic amino acids. In the similar manner, if you see any oxaloacetate or acetyl-CoA, these are regarded as ketogenic amino acids. So, here aspartate is also aspargine also ketogenic amino acids. So, in this manner, based on the end product, so if you see alanine, cysteine, glycine, hydroxyproline and serine, these are glycogenic amino acids, as these can be converted into the end product pyruvate. So, hope you have understood about this. So, if you see the diagram, arginine, histidine and proline, these are converted into alpha ketoglutarate, which is a intermediate of Krebs cycle, which make it a ketogenic amino acid, and aspartate is converted into oxaloacetate, and phenylalanine and tyrosine are converted into fumarate. Isoleucine, methionine, threonine and valine are converted into succinyl-CoA. So these are the intermediates of TCA cycle which are helpful in the formation of this ketogenic amino acids. On the other hand, here glycine, cysteine, alanine, hydroxyproline, serine, asparagine, threonine, these are glycogenic amino acids. Okay, so this is about ketogenic and glycogenic amino acids. In the next class we'll see about the creatine synthesis and other reactions. Okay, thank you students. Please write the notes. If you have any doubts, please contact me. While drawing the structures, while writing the structures, uh, be very careful and write with utmost care. And if you have any doubts, please contact me. Thank you.